right, good morning, everyone. So as I said, my name is Alyssa, and I'm the new Field Crops Extension Plant Pathologist with the University of Delaware. I am located at our Georgetown station, and I started last October. So today I'm going to be walking you through the first year, some of our initial projects and where we're headed with research, along with some of the diseases that we saw this season. I do cover field crops, so small grains, corn, soy, and also do some research in a new crop called stevia. Starting out, when I got here last October, I guess I was hanging out with the entomologist too much because one of the first things I started doing was counting aphids. And the reason we were doing this was for Barley yellow dwarf virus. So I'm going to kind of start today's talk with wheat, then we'll move through some corn and soybean projects as well. So with Barley yellow dwarf virus, this is vectored by aphids. And in the springtime, you then see this yellowing or purpling of the wheat. And the project that we were doing here and why we were counting aphids, we were looking at some seed treatments that we had put out with an insecticide or not, looking at aphid level, and then trying to correlate that to amount of Barley yellow dwarf that we saw in the springtime. This one's a pretty patchy disease, and so come springtime there was a little bit of Barley yellow dwarf virus around, but really not enough to be able to get any rating differences among our treatments. I will make a note with this one, it is important to remember that with viruses, our fungicides aren't going to work. And this spring, there was quite a bit of aphid movement and a lot of aphids around. But with Barley yellow dwarf, usually that transmission is happening in the fall. So when we think about getting our wheat in the ground and planting, early planting is great, but that does leave a wider window sometimes of where you might have more aphids around. And in the springtime, even if you're seeing a lot of aphids, chances are they're hopefully not contributing to the Barley yellow dwarf virus accumulation in the springtime. And with this, you can also sometimes get some purple firing of these tips. So it's called Barley yellow dwarf virus, but you can also see purple and some other colors that come along with this one. I'll spend just a few minutes talking about powdery mildew. This is a disease that I don't have any active research in at the moment, but just wanted to mention it because we do often see it in our area. This is a fungus, and what we see are these white spores, kind of give it that powdery look. And typically with this one, you may get some yellowing of the leaves, but hopefully if you have a resistant variety out there, you're not seeing too much. This bottom image shows kind of the importance of resistance with this one from a susceptible to a resistant, just the level of amount of pathogen that you're able to see on there. And when we think about the disease cycle of this one, we start seeing it become active when we start getting into temperatures over 60 degrees for a week or two. They like that warm spring weather, so a lot of times if you're out there checking, thinking about nitrogen applications, it's a good time to check and see, how much powder you have out there. The good news is usually this one slows down once we start getting hot, dry weather. So in most years, this isn't something that you necessarily need to put anything out to control. And kind of where I'm heading with this is, we have some early season things that may be flag leaf issues, but a lot of times if you can wait, if you are thinking about putting something out until that Fusarium head blight timing, in our region, that is really the one that is our biggest concern as far as managing. And with Fusarium head blight, this is caused by a fungus, Fusarium graminearum, that's able to produce a couple different types of spores. And what happens are these spores land on the wheat head at flowering, and they begin to infect. And so we might see, some hits in yield, but really the bigger thing we're worried about with this one is the mycotoxin that's able to be produced by this fungus. If you've heard, DON or vomitoxin, that's the fungus that's coming from this pathogen. So within this one, as I mentioned, there's a couple different spore types, and these land on the wheat head, and it's important to consider, kind of where these spores are coming from. So a lot of our wheat we're planting in corn fodder, and corn is also, host for Fusarium. So as it turns out, we have that corn fodder there, it's sitting all winter. That allows some of these spores to kind of form, and then when we get into the right environmental conditions in the springtime, this is really favored by rainfall around that wheat head flowering. Those spores will release and come up into the wheat head. in plant pathology, we talk about disease cycles and disease triangles. And so with this disease triangle, what that is really bringing together the factors to cause disease. And in the case of Fusarium head blight, when we talk about hosts, we don't have any true resistance. We have some partial resistance, and I'll mention that a little bit later. We definitely have the pathogen around. It's in our corn systems, it's in our field histories. And then the other key piece of this becomes the environment. And this one is favored by rainfall. And through Penn State, we have our Fusarium Head Blight Prediction Center. And this is a web page that you can access. They update it weekly and daily in the springtime. And you can sign up to actually have updates emailed to you or texted to you. And some of what we end up seeing here is this tracking of green, yellow, red. And so, pretty much for the past couple years, by the time we're at springtime and wheat is flowering in our zone, it's just red across all of the mid-Atlantic. And that is because we've had a lot of high rainfall springs that are favoring disease. And what this is telling us is, it's not necessarily a guarantee. Like, your field may not get hit with as much rain as a few minutes down the road, but based on weather patterns and conditions, this is just letting you know that you're at high risk when you're in that flowering period. So then, that kind of helps to think about how you're going to manage this and if you're going to make the decision to put a well-timed fungicide out. And I'm going to spend the next bit talking about some of our fungicide trials, but before we start in that, I just want to kind of make sure everyone's oriented when we talk about fungicide timing, kind of what I'm talking about here. So one of the timings is 10.3, so that's when you have about half the head coming out. When I use the term anthesis, that means flowering, or you have those nice yellow anthers on the wheat head. And when we talk about post-anthesis, then those yellow flowers are now more of a white. And all of these timings are going to come into play when we start thinking about putting our fungicides out. So I had two trials this year, and this is the first one I'll share with you. This was planted in Shirley, susceptible to Fusarium. And we were looking at the new product, Miravis Ace, and comparing with Prosaro. And with Miravis, it is labeled to have a wider window of 10.3. So we were looking at that 10.3 timing, anthesis, 4 days after, kind of a combination, and then Prosaro and no fungicide. And in this graph, what I'm showing is Fusarium head blight incidence and severity. So when we talk about these, in the blue bar is incidence. So what that is the number of wheat heads that have any sort of symptoms. So I go through each plot, count 40 wheat heads, and see, which of those had some symptoms on there. And what we were able to see here is that, our untreated was way higher, and all of our fungicides were working quite well in managing Fusarium head blight incidence. And so then for the severity piece, within each of those heads that had some symptoms, how much of that wheat head was then covered? And so that's our severity value. And again, we got nice separation between the control and all of our fungicides and timings. So I started in October, and as I mentioned, I'm located in Georgetown. And it's the first time that a plant pathology lab has been in Georgetown. My position has always been located on main campus previously. So part of what I was doing last winter and spring was actually creating a lab and getting to be a general contractor and getting all of the things in the lab to be able to do science. And one of those things that us plant pathologists use are an autoclave. So it's a large machine where you can sterilize things, and that's kind of what we use to sterilize media to grow our fungi on and to make things to inoculate a trial. So at the time of setting these trials up, I didn't really have a functional lab yet. So I just want to make a comment that everything that I was doing in the springtime is just natural inoculum. So I was just, in the field and kind of just waiting to see what Mother Nature brought our way as far as diseases. And one of those things was Stagonospora leaf and Glume blotch. I had intended for these to be Fusarium head blight trials, but ended up with quite a bit of leaf and Glume blotch. This is another one that's caused by a fungus. And if you're ever looking through literature on fungi or trying to do a Google search, people are constantly changing the names of fungi. And this is one where it's gone through a bunch of different name changes. So I'll take a minute just to kind of orient you. So, a lot of people I think in the area know this as leaf and Glume blotch. This used to be called Stagonospora, the fungus. Now it's called Stagonospora, and for whatever reason, there's some articles out there where it's now, for a common name, also called Septoria nodorum blotch. So when you start Googling for this one, you get all kinds of hits, but it all leads back to the same thing. And for today, I'm just going to call it leaf and glume blotch. And this is one that can survive in residue for a couple of years. And it can be both in the leaf and flag leaf, and we can also have issues in the glume. When it's on the flag leaf, we tend to see these cat eye shaped lesions. And then when we move into the glume, it's kind of this purpley brown, and I'll show some pictures later, but when you look out at a field, it just kind of looks dirty. It has kind of this grimy look to it. So with this trial, we rated flag leaves, and what we were doing was a 1 to 5 rating scale, just sort of the level of lesions that were on there, anywhere from 1 to 2 lesions, up to a 5 being the whole flag leaf's just covered and you can't see any more of it. And when we take a look at our numbers, so our control, that flag leaf was pretty much kind of gone, and then we had most of our fungicides keeping it down to that 1 to 2 lesions. So when you have those flag leaf lesions, it's not a necessary guarantee that you'll have the glume blotch, but they do often go hand in hand, and that was the case in our field. So in this one, the glume blotch incidence is in blue, and this control is the very tall bar, so pretty much our whole field was just covered in glume blotch. And again, our fungicides were working well and providing some control there for both our incidence and the severity or amount and level of damage on each of those heads. When we look at moisture of this, we did have a little bit of difference come out, that our Miravis products had a little bit higher moisture. And with this trial for yield, we didn't end up with any significant differences. Numerically, our control was the lowest one here at 61 bushels. And when we talk about Fusarium head blight trials, again, that incidence and severity is important as far as our visuals go, but really the key is what are those DON numbers? When it comes time to go to the grain elevator and we're thinking about being docked or rejected, no one cares like, oh, did you have a beautiful looking crop? It's, what are your DON numbers? And in this trial, it again was naturally inoculated. So in all of my treatments, I ended up with pretty low levels of DON. About 0.3 was the highest in the control. None of these ended up being statistically different, but if you notice, some of these anthesis applications or right after anthesis did end up being numerically the lowest. And this is a trend that we've seen across many states in the U.S. This is a snapshot of some work in Wisconsin, and if you just focus on that DON column there, what we've been seeing is that some of those earlier timings have a little bit higher DON levels than the anthesis or a little bit after. Taking a look at some of the work in Kentucky, again, the 10.3 a little bit higher than at 10.5.1. And really, when we start thinking about this and what it comes into is when timing is still really important with this. And 10.5.1 is probably still the best timing to have. Because when you think about that, it's your main tillers are at 10.5.1 or a little bit beyond, but you still have all those secondary tillers under there. So the goal here is, hopefully if you're hitting your main tillers at 10.5, then maybe those secondary ones are about 10.3, so they're hopefully getting picked up as well. Whereas if you spray all the main tillers at 10.3, those bottom ones may not be open yet and not getting as much activity there. So when we think about Fusarium head blight, timing is still really important, and that 10.5.1, or when you have those nice yellow flowers out at anthesis, is a good time to apply. Had a second trial looking at a few different combinations, some of the same treatments as that first trial, and a few others, and then this one was a more resistant variety. And with Fusarium incidence and severity, overall our Fusarium was a little bit lower, but the control was still, by and large, very separated out from all of our fungicide treatments, and same with our Fusarium head blight severity. I had mentioned the glume blotch of looking dirty, and this is just kind of that visual example of I was out in the field raiding, standing in this wheat, and you look over, and it's like, wow, this is, quite a dramatic difference between our glume blotch plots and the ones that didn't have it. Luckily, I work with someone who's got a drone, so Jared took some pictures for me. And in this little square here is where I had this wheat trial. And all of these bright patches are where we had fungicides that were having efficacy on our Glume blotch. And so that you can see it kind of split out between treatments here. And to put some numbers to that, again with the Glume blotch incidence and severity, what we were seeing is that our control was near 100%, so like the entire plot was just covered in Glume blotch. And we weren't getting as much activity out of Prosaro for Glume blotch management as we were out of the Miravis products. And within this, again, you can see the orange severity, same trends going along there. With this trial, again, with moisture, we did see a little bit higher levels with the Miravis Ace products. And this one with yield, we did have some differences come out. So this one didn't have quite as much Fusarium head blight as that first trial, but a lot of Glume blotch. Again, this was naturally inoculated, so it was just things that happened to be out there that year. And our top yielder ended up being that one that had 2 applications, but that was followed by some of these other single applications of Miravis Ace. And so again, when we talk about yield and our incidence severity type of things, we're not seeing a lot of differences in these timings, but the one area that you do want to consider is for those DON numbers, still kind of going with that recommended 10.5.1 timing with these. So when we think about developing, integrated wheat management program, we have things like barley yellow dwarf where we want to consider planting date. We want to think about our rotation. A lot of times it's tough to be in anything other than corn. But this year there were a number of fields where beans got off early, so if that's an option. But if if you do have that corn out there and you're coming in with wheat or barley after, then thinking about your fungicides, but also thinking about your varieties is important. And through the University of Maryland, they run a program where they do a misted nursery. And so what this is really extreme environment. So it's testing these varieties against like the most disease you could ever have. So they're inoculating really heavily. They're putting a lot of moisture and misters on. And so in the end, you end up getting these really high DON numbers. So I know it's scary to look at these tables where you're like, oh my gosh, DON of 15, like what's going on? But again, these are like really extreme. And so what the idea here is you're trying to separate out varieties so that the ones that have the lowest amount of DON in this extreme situation would hopefully also be the ones that have the lowest amount of DON in a naturally inoculated setting. And this is just a snapshot of their table from this year. You can find that online. And one of the things that we'll be doing next year to kind of parallel with this over at UD is actually in our variety trial, I'm going to be doing some ratings and some DON numbers on uninoculated trials. So we can kind of compare between these really high disease systems versus a natural system. Are we getting some of the same trends? So kind of wrapping up into 2020 with small grains, we'll be continuing to do some work with Fusarium. So again, that's the causal agent of head blight. It's assumed that it's mostly just this one species, Fusarium graminearum, but there can be some other Fusariums that can be involved as well. So we've started kind of an isolate collection where we're looking at what kind of diversity we have around here. We'll be continuing fungicide efficacy trials next year. And we, relied on natural inoculum this year. Next year, I'll be doing some misted trials to put a little more disease pressure, but we'll keep some natural inoculated trials as well. And I also will be starting some trials in malting barley next season. I did some scouting through malting barley this year, but just didn't get here in time last year to really get any trials started. With malting barley and barley in general, for Fusarium head blight, the timing's a little bit different. You do kind of want it to be, it's a little later, you want to be at that full head. And so hopefully this time next year I'll have some numbers for you on some malting barley timing. All right, any questions on wheat before we kind of jump into corn season? Okay. Well, this spring it started out pretty wet, and I was a little worried we were in store for another 2018 of just wet all year. But as it turned out, there was actually, kind of faucet turned off and it got pretty dry. And really with a lot of our corn foliar diseases this year, we didn't have some of the levels that we saw for last year. But with that wet spring, what I saw a lot of was damping off. And just by a show of hands, did anyone in here have fields they were in that had damping off issues this year with corn? Couple. So I got called out into quite a few fields, and in most of the cases, these all had beautiful stand emergence. And then it was about 3 weeks after we start seeing this post-emergence damping off. And when you go to dig a plant out, they're kind of brown. A lot of our roots are dead. In many cases, the top was like completely separated. If you go to pull it, you couldn't even get those roots to come out. And what we were finding was a lot of different species of Pythium. And so we collected a lot of isolates this spring, and I actually have a master's student that's going to be continuing into some of this work. And just very hot off the press, this is like 3 days old, we've been getting some sequences back of some of the species that we actually did have out there. And just in the first 20 or 30 samples that we've ran so far, we've had over 6 different species of Pythium. And with Pythium, the thing to keep in mind with these species is that some are favored by cooler weather, some by warmer weather. So this year when I got called out into these fields with damping off, I was in some fields where they had planted early, had damping off issues. I was in some fields where they had planted late, still had damping off issues. And probably what's going on there is that you have different Pythium species that are favored by that cooler or warmer weather. And so with this project, we're going to be looking at some of the aggressiveness of these different isolates, looking at some in vitro fungicide sensitivities, and also some seed treatment work out in the field. So we're pretty excited, lots of directions that we'll be heading in understanding this Pythium in corn. And also, kind of connecting it to then late season, we also saw a fair bit of stalk rot this year. And it did seem that some of these fields that had a lot of early season Pythium were also some of the fields that had high levels of stalk rot later in the season. So we have a few projects set up where we'll be tracking plants through the season and seeing if that early season Pythium is connected at all with some of that late season stalk rot. So in addition to some of our Pythium work, we do a lot of fungicide trials. And if you've never seen the research size corn boom, my friends always laugh and joke with me that I'm like trying to fly to the moon but hang my laundry at the same time. It kind of swings behind you, and you can get over and spray these center 2 rows. And with these fungicide efficacy trials, what we're going for is our foliar diseases in corn. And around our area, usually the big 2 we're worried about are gray leaf spot and northern cornleaf blight. With gray leaf spot, this tends to be a very boxy lesion that follows the veins. Of course, as with anything, there's exceptions to the rule, so you can sometimes see some variation out there, but usually that one stays pretty boxy. And with northern cornleaf blight, it tends to be more of an oblong, kind of cigar-shaped lesion. In previous years, we've had quite a bit of northern cornleaf blight around, but I really didn't see a whole lot this year. Mostly the fields and samples that I was getting were gray leaf spot. And a lot of that was, we were pretty hot and dry through most of peak corn time. So I actually ended up with 2 trials out this year. The first one that was planted in May got really heat stressed and didn't get any disease. So I'm not going to share the results from that one. But I did also have a late season trial that ended up with a lot of gray leaf spot. I'd actually planted corn getting ready for some wheat trials for next year and just noticed how much gray leaf spot was out here. So layered in another fungicide trial. And so we were looking at a variety of different fungicides, of different modes of action and groups, and all of these were applied at R1. And so in this field, at the time of application, we already had pretty good disease started in the low canopy. Most of the flag leaf or ear leaves in this one were at 2 to 3% disease severity. And when we talk about disease severity with corn, it's looking at that ear leaf and the amount of leaf that's covered in lesions. So to take a look at our gray leaf spot ratings, we rated at a couple different time points. So up here I've got 14, 28, and 35 days after application. Down here on the bottom you can see the control. So by 14 days, we were at almost 20%. And by the end we ended up about 71% of that leaf area covered in lesions. We did get some good separation, and many of our fungicide products were working better than the control. Again, this is a pretty high disease pressure within this environment. When we look into yield, because this was so late planted, there just wasn't a lot of yield potential in this corn. So we didn't end up seeing any significant differences in our yield, despite the really good control as far as the gray leaf spot went. And then just to kind of bring that back together, Again, the control down here at the bottom. All right. In addition to kind of traditional, fungicide application trials, I've also ended up in some projects with James over at our Warrington irrigation farm. So before I got here last year, James and Corey started, doing a little bit of kind of a proof of concept last year of what happens if you put, fungicide in the irrigation system. So we started this year with looking at, we had about a 90 by 90 block where we had a susceptible plant and a resistant line, and we were looking at no fungicide, kind of a premium, like three-way, so hitting it with the kitchen sink, and looking at V8 and VT timing, VT and VT in about 14 days after. Then we also had tilt with a single mode of action, again looking at those same timings. So, excuse me, in this graph, in our blue we have susceptible line, and the orange is our resistant line. And so, one of the first things that you can notice here is like the value of planting a resistant line. So just by planting a resistant, you can eliminate, a lot of your disease pressure. In this particular graph, we're looking at gray leaf spots. So again, it's that ear leaf and how much of it was covered in lesions. Within this susceptible, we were seeing good disease control, especially, with some of these applications of V8 and VT. So this is pretty promising to us that, when we think about putting fungicide out through this irrigation, it's a really high carrier volume. So one of our big questions was, if we put this out, how much of the fungicide is actually making contact and staying with the plant versus just getting washed away? But we were seeing some promising results here as far as disease management. In this field, we also had another disease, Diplodia leaf streak. And this one can look very similar to northern corn leaf blight, but inside it has some little black structures. And, this is another one of those cases of where they changed the name, and, in my opinion, not for the better, because this used to be called Diplodia, and it has these spores that, have two sides to it. Makes sense, but they changed the name, and so now it's called Stenocarpella macrospora, so it doesn't connect as well with the spores. But this is one that you can see it in this leaf streak stage. It can also be present as an ear rot, and it also can be present as a stalk rot. And there was a lot of Diplodia stalk rot around in fields this year. I didn't see the leaf streak phase in too many fields outside of this one in Warrington, but we had quite a bit of it there. And if we look, again, at our susceptible and resistant, again, you can see the value in having a resistant line. With the susceptible, our fungicides were helping. With the resistant, it was already low enough that even with the fungicides, we weren't getting any difference because it was already really low disease level. So with these, we had these large 90 by 90 blocks, and we kind of then cut out the borders and harvested the centers of each of these plots. And this is data that we're still in the process of analyzing. So far in what I've seen, we're getting some numerical differences come out in yield. So between the response that we've seen and actually, reducing some level of disease and the yield that we've seen so far, we're going to continue this project a second year and work on optimizing, there's a million questions that we have as far as, what if you change nozzles, or what if you change, surfactants used, rates, and so we've got a million questions, and so this year was kind of just a starting point, and we'll hone in on further optimization in future years. So next year, moving into our corn research plan, we have lots of projects going on with Pythium, again looking at that fungicide aggressiveness and sensitivities levels. We'll continue our efficacy trials in field corn settings, also looking at that irrigation system. And then I don't have time to share it today, but also did some sweet corn variety trial rating for gray leaf spot as well. And that'll be some information that I share at Delaware Ag Week in a few months. So any questions with corn before we jump into soybeans? We're going to wrap up today talking about soybeans. We had a couple different projects going with soybeans. We were doing some double crop row spacing and fungicide timing trials. We had some full season fungicide trials. One of the big things where I'm going to focus on today is establishing some areas for nematode research. And we were doing a survey throughout Delaware and Maryland looking at nematodes as well as some of our soilborne fungi. And this year turned out to be a pretty good year as far as from a farming perspective. There weren't a lot of soybean diseases out there. There was a little bit of early season Septoria low in the canopy. There was some SVNV around, but by and large, there were very few fields that I got called out into. I heard a few reports of maybe some SDS things here and there. And then the stem canker, which is the diaporthy, one of those that we're looking at, there's a little bit of that around as well. And we had a couple with macrophomina or charcoal rot. So with this survey work that we were doing, this is just sort of a snapshot to show you what it looks like inside of a lab. So we go out to the field, we collect all these plants, and we bring them back. And these are all petri dishes where we have different fungi growing. So once we bring these back to the lab, we're then able to do some follow-up work of seeing, learning about them, knowing what species we have. There's many cases of where, things in the field, they can all look pretty similar, but until you start looking at some of the molecular level, it's hard to know exactly how different some of these things are. So this is where we do a lot of our fungicide trials in vitro and plate assays and learn more about our pathogens. I don't have time today to talk too much about our fungal pathogens, but what I would like to focus on is soybean cyst nematode. So when we look at the most damaging soybean pathogens across the U.S., SCN usually consistently ranks as number one. And the thing with soybean cyst nematode is a lot of times it's there and you may not even necessarily know it's there because you get stunting, you get things that aren't necessarily visually striking, but they can really inhibit yield and be quite a yield reducer. SDS ranks up there. We have some years where we have it, some we don't, but our zone we certainly have a lot of the seedling diseases around as well. So when we talk about soybean cyst nematode and why it consistently ranks number one, there's sort of two prongs to this. So one is that soybean cyst nematode numbers can increase really rapidly. So these females will attach onto the soybean root, and then inside of each one of these females, they're able to produce around 250 eggs. So if you can imagine, you have a couple hundred females on a plant, and then each one of those has 250 eggs. You can really get some exponential growth very quickly with these. The other problematic thing here is that SCN can survive well in the absence of hosts. So these are a couple reports of, being 40 years later, you can still find it. I find ones like that kind of depressing because, man, that's hard to set up a trial for. And there's another one here where 8 years of fallow and then susceptible grown in years 9 and 10. And so by that second year, it's already an issue again. So this one is, very troublesome. And really where a lot of this trouble comes in is for years we've relied on resistant varieties. So this is a picture kind of showing a susceptible and a resistant. So this is awesome because, really you don't have to change anything here. If you're buying seed anyways, just buy the resistant one. And this is like a $0 investment. You get great yields. But our trouble that we've been seeing is that, our main source of resistance in soybeans is this PI 88788. And it's in, over 90% of our soybean lines. And this is some data from Iowa showing the amount of reproduction. So the idea of this resistant line and what the goal is there is that you're not getting reproduction. So the nematodes, latching on, but instead of getting those 250 eggs out, you don't have any. But what we've been seeing is this slippage. So the goal with these resistant lines is that reproduction should be less than 10%. But as you can see, pretty much since 2000, that number keeps creeping higher and higher of where instead of 10% reproduction, we're up in the 60, 70% reproduction. And, there are some other lines out there and sources of resistance other than this PI 88788. But, sometimes they're a little harder to find, and again, it's only making up probably, 5 to 7% of the market, whereas this one's in everywhere. So a few years ago, there had been this SCN coalition that existed, and then they kind of revamped it a few years ago. And one of the things that they've been trying to promote is just help people have awareness of, thinking about, because for years we just didn't really have to think about what our resistant source was. But, if you're thinking and take a look at your seed, see where your resistance is coming from. It's probably this PI 88788. But we are seeing that loss. And because of that, if you start having nematodes over your whole field, it can be really difficult to know that you have them there because If the whole field just looks stunted and you don't have anything else that's very visually striking, then you could, think like, oh, it's just the weather, or that was the only yield potential there. So for this year, one of the first things that I was interested in doing was just sort of getting a survey across Delaware and Maryland of what kind of numbers are we seeing and where are we finding nematodes. So in yellow, these are all fields that had soybean cyst nematode. Blue didn't have any. Red were these root knot fields. And orange had both soybean cyst nematode and root knot. So I was in about 60 fields. A lot of times, especially for these SCN fields, they just looked like normal fields. Like, you look out and there wasn't necessarily like a problem area. And in most of these samples, I was just taking a field representative sample, but getting really high numbers of SCN. It was interesting, these root knot fields, those ones did sometimes have some visual clues of, it's just some areas of the field where they did look off. So with root knot, you can sometimes get a little more help in thinking about what those symptoms look like. But a lot of times with SCN, it's just sort of silently there and something that we have to deal with. So my goal in kind of starting this project this year is just having a handle on where we have some SCN hotspots and trying to get some trials started looking at some seed treatment options, looking at maybe some of these other sources of resistance and seeing if we can get any success with those. So moving into next year, I'll be continuing some of this nematode survey work. If you have a spot where you think you have a hot spot, feel free to contact me and be happy to try to include it into our surveys if we're able. Again, looking at some of these seed treatment trials. And then we'll keep looking at some of our full season fungicide efficacy trials for our foliar pathogens. And again, understanding some of these soilborne fungi. In our zone, we often have both northern and southern stem canker. So trying to understand, Diporthy is the kind of scientific name of that, but understanding some of our northern and southern stem cankers, some of like the Phomopsis seed rot, and some of those are in our program, as well as Macromina or charcoal rot. And I will make a note, if you have interest in SCN, I do have some handouts up front, so feel free to grab those at the end. I just wanted to wrap up by thanking Delaware and Maryland soybean boards, along with the Maryland Grain Producers Utilization Board, as some of our initial funding for these projects. And then a shout out to my grad students that our lab's slowly growing, and you'll hopefully be seeing more from these folks as they're working in corn projects, soy projects, and hopefully have another coming on with wheat projects. So with that, I'll be glad to take any questions that you might have.